Great. So we'll just leave it a couple of moments longer. I can see we've got a fair number of people joined in, but before we do, just going to explain how the session's going to work. If you have any problems, if the audio is breaking up or anything like that, let me know in the chat. If you've got any questions as we're going along, pop them in the Q&A, and I'll ask them to Gareth at appropriate moments. I'll let him come to a stop and then ask him something. This session's been recorded. I'll send a link afterwards, the same I did with the sessions yesterday. But it looks like everyone should be joined in now. So I'm going to hand over to Gareth. Over to you, Gareth. If you'd like to introduce yourself first of all.
Sure, I'm Dr Gareth Mannall. I look after the facilities here. I manage primarily the pilot plant, and I have experience in using a lot of the kit that I'm going to show you this morning. So without further ado, I'll just change my camera, hopefully, and so instead of looking at me, you can look at equipment. So hopefully that will work. And I have my finger in front of it. So I want to start off in terms of the very first stages of our process. What I'm going to do is take you around around our facility and show you what a bioprocess looks like. So the very first stage that we start with, if I just take us towards where we've got some algae growing, okay? We grow algae, we grow bacteria, we grow things like mammalian cells. And typically those things are going to be producing proteins of interest, okay, or typically proteins of interest, things like therapeutics, things like vaccines, okay. So the very first stage that we're going to do is we are going to grow these things at small scale, and typically what we use are these shake flasks. So you should hopefully in view now see what look like conical flasks to say. So in those conical flasks we have nutrients that the cells need to grow, and they're being shaken to provide them a bit of air. So they've got the kind of the carbon, the nitrogen and the oxygen that they need to grow in, and they're being mixed to make them grow slightly better and better. So that's the very first stage that we can do, but typically we can't scale like that. So typically when we want to produce grams, kilos, tons possibly of material, it's not possible to grow them in small shake flasks like that. So we need to be thinking about a means that we can scale up, okay. So the only means of control I've actually got in my, I don't know how well this is going to come through on my phone, and in terms of my little shaker that I've got here, is how warm I keep it and the temperature that I keep it at. The cells are going to be reliant upon other things like the amount of oxygen that they have, and the pH to allow them to grow properly. So what I'm going to quickly do is I'm going to pop downstairs and show you a fermenter. And a fermenter is the instrument that we use to grow organisms at scale. So bear with me. I'll swap my camera around so you can just see me whilst I walk down the stairs rather than the stairs. And I'll try and make sure I don't. I watch my step as I come down the stairs. Okay. So if there's questions along the way, let me know. So I'm just going to quickly swap my camera again, and I'll show you what a bioreactor looks like. So hopefully in front of you and on your cameras at the moment, you can see a stainless steel vessel called a bioreactor. Okay, so this is a bioreactor, and it's a stirred vessel that we use to maintain an environment that the cells are happy in growing in. So what I talked about was what things do we need to allow the cells to grow properly. So they need to have the air, the nutrients. So in terms of kind of nutrients that we provide these things with, we need to provide them with nitrogen, and that source of nitrogen. This can come from things like yeast extract or proteins, but hydrolyzed milk proteins. They're going to need a carbon source. So typically give them sugars, things like glucose. We can give them complex alcohols, things like glycerol. And that's the kind that gives us the carbon and the nitrogen that they need to grow inside the reactor. They also need to have oxygen as well, and that is provided by sparging air into these reactors. So usefully, one of my colleagues has slightly taken apart one of our bioreactors that sits next door. So what you can see is inside the bioreactor. You can see what it looks like inside. So you can see, hopefully see stirrers. You see this kind of shaft that we've got, And that thing that looks a bit, I suppose, got 5 blades on it, that is the stirrer that sits inside the bioreactor and helps to keep things mixed. So instead of shaking them, we're stirring them, okay? And inside there, I don't know if I can actually see right at the bottom, is something that looks a bit horseshoe shaped, okay? And that is, as horseshoe shaped, is the sparger. And that sparger, it bubbles air into the bottom of this reactor to provide the cells with the oxygen that they need to grow. So we charge this vessel with the media that provides the carbon and the nitrogen they need to grow, and then we sterilize it, okay? So typically for sterilization, we use steam that gets put into the jacket section, which is here, okay? And heats the contents up to 121 degrees. Many of you don't probably use pressure vessels or kind of pressure cookers or anything like that nowadays, but that's what it does. It gets the temperature up to about 121 degrees. Heat gets it there for about 20 minutes, and that sterilizes it. Then what we can do through the addition port on the side, if we grow our cells up like in those little shake flasks that we saw earlier on, into this bioreactor, we can inoculate the bioreactor with those cells, and we can grow the cells within the bioreactor. So during the fermentation, we need to control, okay? So we need to look at the temperature. So that jacket is also used for temperature control. We'd look at the pH. So you can see 2 probes at the bottom here, hopefully. There's one that's got a little red bit on, that's our pH probe. The one that sits next door is the DOT probe, okay? So typically our cells, something like E. coli, is happiest around a neutral pH, that's pH 7. Well, as the cells grow, they typically produce acid, and we need to counteract that acid. So what we have is a means of pumping acid and base into the reactor to maintain the pH. As I say, a lot of the time, because it's typically the cells are going to secreting acids, they grow, we're putting in base. We're putting something like sodium hydroxide or ammonium hydroxide to control that pH and bring it back down to the pH that we want them to grow at. As well as that, we need to provide them with the oxygen they need to grow, and that will be provided by sparging air through the bottom. Once air doesn't become, so we'll be monitoring the amount of dissolved oxygen inside there. Once that doesn't become enough, we can either sparge more air, or we can increase the speed of that little stirrer that I showed you earlier on to break up bubbles better and provide better mass transfer so the cells can grow better. Typically for our microbial fermentations, we tend to do a bit of growth, and then we'd add something to actually make the cells that we're growing produce the product of interest. So there'll be some sort of chemical that we add. Once we add that, then the cells will probably start to shift their metabolism a tiny bit. So what happens? They go from mostly growing, or complete all the energy, carbon, nitrogen, oxygen is going towards growing. They move away from that towards producing the product of interest, be a vaccine, a recombinant protein, something like insulin, an anti-cancer drug, or something like that. So that's the kind of things. This is the kind of vessel that we grow cells in. Okay, so the typical kind of vehicles we use for production are, as I said before, things like E. coli, a yeast called Pichia pastoris, and sometimes we use the mammalian cells for production because they can produce. Because they're more similar to our own cells, they produce products that are more similar to the human equivalent. Okay, so that is pretty much fermentation. And what fermentation is achieving, what we want to do in that process is to produce as many cells as we possibly can, and hopefully we've produced as much product as we possibly can. Okay, so I'm just going to swap over again back to me. Just as I'm moving around, I'm going to just show you a bit of different kind of bioreactors that we've got. Hopefully I'm not actually necessarily seeing it. Sorry, got a bit dark. I'm not necessarily Q&As, but I'm happy to answer stuff as it comes or as required. So what I'm going to quickly do is swap my camera around again and just show you very quickly, just simply some of the smaller bioreactors that we use. So we can go a lot smaller with our bioreactors as well. So if we are looking to optimize these conditions to produce as much product as we possibly can, we might want to do many conditions in parallel. So this kind of bioreactor we've got here is a 4-way bioreactor. There's 4 little bioreactors. It does everything the big one does, but on a smaller scale. And that means it's cheaper for us to do, and we can do multiples at the same time. By doing that, hopefully we can get to some sort of optimum condition. So I'm just going to move around. The largest bioreactor that we've got is the 100 litres. The one that I showed you earlier on was a 30 litre. This is a 100 litre. This is kind of like a proper pilot kind of scale that people probably use in industry, but in industry, you'd be growing possibly to 10,000 litres, possibly 100,000 litres. So this is relatively small, but it's about as big as we do. But these are very predictive of the large scale kind of fermentations that we might do at scale. Just very quickly, because it's going to be a little bit easier to see, some of our bioreactors, instead of being stainless steel, they're glass. So that means instead of sterilize them by putting steam into them or heat them up to 121 degrees, we simply put these machines into autoclaves. An autoclave pretty much does the same job. The autoclave heats up to that temperature and sterilizes the contents of the bioreactor itself. Okay, so that is pretty much what we call upstream. That's where we produce our cells, and hopefully, if we've done everything properly, our cells should be producing as much product as they possibly can. Okay, when we do grow thing in mammalian cells, the mammalian cells grow in slightly different bioreactors. I'm not going to be able to show you those today, but those bioreactors, because the cells are more sensitive to shear. You may remember seeing those little blades I show you on the stirrer. They use a different design that's slightly softer, so not harmed by the stirrer speed. Okay, so for a mammalian cell process, you want to have a slightly gentler approach because those cells are more sensitive to engineering parameters. Okay, so hopefully after you've grown all these cells, we need to typically either our cells produce the product it will excrete the product out of the cells, or very typically if we're doing bacterial kind of stuff, what will happen is those cells will produce the product inside the cells. So regardless of the process, the very first thing that we need to do is to separate our solid away from liquid, okay? So I don't know how much centrifugation you guys have done, but very typically the kind of stuff you do at small scale would be in a centrifuge much like this, okay? And what you would do, you'd put in a tube, much like this, okay? And you spin it at high speed to separate the liquid away from the solid. Okay, so hopefully you can see that. So I was holding that slightly out of camera. So that's how you separate solid away from liquid. Imagine, if you will, we have got tens of thousands of liters. We can't get really big. It'd be like almost like a fairground ride in terms of the kind of size of the thing that you'd be rotating to separate the liquid away from solids. That's not possible, okay? We can't do that. So that instead, what we can use is essentially one of these machines or a similar machine, which we call a semi-continuous centrifuge. Here's the difference. Instead of actually chucking batch of material into the machine, what we're going to do is we put liquid onto a rotating bowl, okay? There's different designs. This one that I'm showing, is called a disc stack. Us being engineers, we're not particularly creative in terms of naming of stuff, but it's simply the separation area is a stack of discs, okay? So we flow liquid in because the bowl is rotating at high speed. This one here rotates about 13,500 RPM. The solids, being more dense, will deposit themselves inside the bowl and the clarified liquid comes out. So on this machine, the tube that's coming from the top, that's where the liquid comes in. It then goes through the centrifuge, solids deposit themselves inside the bowl, and a clarified liquid is collected, okay? And we can, with a machine like this size, 1 litre bowl, we could do about 200 litres an hour. These things get bigger and bigger. I was talking to a colleague, or actually one of our PhD students who's been working in industry recently, and he said that he was working with a machine that was 10 times its size that pretty much filled up a room. So this is kind of machine that we do for solid liquid separation. The problem with these machines, well, it's not a problem, I suppose, that once they fill up with solid, they no longer separate solid away from liquid. So on this design, I don't know if you can hopefully see this picture that's in front of you. This just shows you a bit of details of how it looks like inside, almost like a cross-sectional view. And what happens on this device is that I'll try and start on the first picture. And what you should see is that we start to put liquid comes in from the top, and then the clarified liquid will try and make its way out. as we go along, what's going to happen is the solids will deposit themselves, will come down the discs and deposit themselves here, okay? Then that area is going to fill up with solids. Once it fills up with solids, we need to get rid of those solids. And what happens is that water goes underneath the bowl and pushes the bowl up to expose a hole on the outside of the bowl. By exposing the hole on the outside of the bowl, the high G that's inside the centrifuge sprays those solids out. Once those solids are out, the bowl closes once again, and we can separate solid away from liquid. So that is the kind of solid liquid separation process. There are different designs. This thing here is a tubular bowl, and the bowl, I suppose, is a tube. And the solids deposit themselves inside the bowl. Unlike the one next door, it's got a little scraper in it that enables it to scrape the solids out and get rid of the solids, so you can separate solid away from liquid. Okay, so what we've got in our process, say we're interested in something that's inside the cell. We've got the cells, we pelleted them out, they're separated from the liquid they've been growing on. We need to break the cells open. We resuspend them in some sort of buffer that they're okay in, typically around a pH or the salinity that would be helpful for them. And then we put them through something called a homogenizer. And this thing here is a Lab 1000 homogenizer, okay? So what we do, it's like a glorified pump, I suppose. We put material into this hopper that's at the top, okay? And inside this is 3 components. So it goes into this assembly at high pressure. The kind of pressures we're talking about are about 1,000 up to 1,000 bar in this machine, okay? So we go for a small orifice at high pressure, and that high pressure causes that suspension of cells for the cells to break open and release their product, okay? So it comes in full cells and probably comes out of those broken open. You might need several passes, but once you've done that, you've released your product into liquid, and what you're left with is cell debris. So that is homogenization. Other methods are available. We could use chemicals to break open the cells as well, but very commonly in industry, we use homogenizers. This is, again, we're pretty small scale here. This thing can do about 22 liters an hour. You can get ones that probably do about 100 times that amount in industry, okay? So that's a homogenizer. After homogenization, what we need to do is to separate our products of interest is in the liquid phase, but we've still got some solids left. Those solids are the cell debris. So we've got the cells, you break the cells open, what you're left with is fragments of cell wall and membrane and stuff that you need to clear. So again, we can use the centrifuges that I showed you on, be it a tubular bowl, or the distack centrifuge. But the thing is, I don't know how much you guys know about centrifugation. The efficiency of centrifugation is dictated by how big and how dense our particles are. If we've gone from full cells, we're a bit like balloons, to fragments of balloons, if you like, it's going to be a lot more difficult for these centrifuges to separate the solid from the liquid. So we might get to like late 90s in terms of the amount of clarification and clearance we get, but we need to probably do another step. So that other step is going to be some sort of filtration. So I've got, walking around a pipe, trying to find bits and pieces to show you. Apologies for the background of this, but this here is what we call a depth filter, okay? So depth filter, as its name suggests, it's kind of a deep filter. What it does, it's almost like a little bit like a sponge. You put liquid in from the top. Inside here, it's a little bit difficult to tell. There's discs of depth filter material, okay? So liquid comes in, and it goes through this the depth filter material, and the clarified liquid will come out at the bottom of this device. And what the depth filter does is a bit like a sponge, okay? So the solids, if they're bigger than those pores, they're not going to go through. If they're roughly the same size as the pores, they're going to get trapped in those pores, and they're not going to go through. So what you do by doing depth, the stuff that's left over from the centrifugation, the solids will be removed by this depth filter. And hopefully what you're left with is something that's completely clear of solids, okay? So hopefully by this stage, what we've got is we've got our product of interest, okay? That's in the liquid phase, and we've got rid of all the solids, all the kind of particles that we're not interested in. But bear in mind, we are dealing with biological systems here, okay? It's like a chemical process where we chuck a couple of things in. We might have some sort of byproduct and a product. We've got loads of things we're not interested in, as well as the stuff that we are interested in. So even though we've cleared all the solids away, we've got to get rid of the proteins that the cells need to live, the DNA, the RNA, the lipids, the carbohydrates, all that kind of stuff needs to go away. So what we do is we need to do something called chromatography. So I'll take this off me, and what hopefully you should see is what we call a chromatography column, okay? So that's in front of you. Chromatography column is simply, I suppose, a bed of the particles. It's almost like a bit, Cephros is people typically use. It looks a little bit like agar, it's a bit like seaweed that it's used. Goes into particles about 90 micron, and what we use is we exploit the properties of the protein to separate it away from everything else. So these particles can have things like charge. We can have negatively charged matrix. We can have positively charged matrix. We can have resin that's slightly hydrophobic. We can have resins that bind proteins very specifically. We call those affinity chromatography measures. What we can do, we can use these systems either to bind the products of interest, or to not bind, or bind things that we're not interested in, okay? So in that way, we could do several of these steps in series. What you do in terms of chromatography, you load on your sample and equilibrate it. So what's happening is that everything that doesn't bind goes through, your stuff binds, then you change the condition to a different buffer to elute off your product of interest. By doing several of these stages in series, so one after the other, what we'll hopefully be left with is a product that's maybe 99.99% pure. One thing I suppose, so the problem can be that after this or during this process, we might need to change the liquid, the buffer that the protein is in, either to bind to the column or maybe for its final formulation. So typically the kind of chemicals we might use in terms of purification aren't suitable for final formulation, okay? So I'm going to quickly show you is a tangential flow filtration system, okay? And what this tangential flow filtration system does, it simply allows you to do something called buffer exchange. So unlike the filtration we described earlier on, what we were doing is trying to actually remove the solids by using the filter. On this tangential flow filtration system, what we're doing is we're actually retaining the protein on the feed side. So we use membranes that have got incredibly small holes, typically on a molecular, we call them molecular weight cutoffs. We typically say if our protein, like a monoclonal antibody, is about 100 kilodaltons, that's 100,000 Dalton, we'd use like a membrane that's probably a third of that size, about 30 kilodalton, and we'd use that to retain the protein but get rid, and what we'd use that is we'd filter, we put that liquid on and filter but add new buffer as we go along. By adding new buffer, what we can do is we can exchange the buffer that it was into a new buffer that we're adding, okay? So that's what we call diafiltration. It's a bit like dialysis, I suppose. We're exchanging the buffer that it was in for a brand new buffer. And if we're doing this in the formulation stage, that means we could get into the right salinity and the right pH for the patient to use, or at least the clinician to treat the patient anyway. Okay, so I'm just gonna swap my camera back. I'm almost done, don't worry. About 5 minutes to go. So I think hopefully you guys, if you're kind of interested in like bioprocessing, you're probably reasonably talking about the kind of vaccine stuff and the challenges that have been experienced in terms of actually using the material. A lot of the Pfizer ones you might recall were had to be done in cold chain. Okay, so they had to either transport in very, very cold freezers. Even if we do produce our product as a liquid, the problem is that's probably going to have a limited shelf life, okay? It's eventually going to go off. It's like anything. You have orange juice in the fridge, you leave it for too long, it goes off. This is just very expensive orange juice, I suppose. You've got a very expensive protein, and if that stuff goes off in the fridge, you've wasted it, okay? So what we can do, and I'm just going to show you the machine that's in front of me now, this is called a freeze dryer, and probably the kind of, you've probably heard of freeze-dried coffee. And maybe you may well have heard of freeze-dried proteins as well, hopefully. But we also, the name is also, you might have heard lyophilization, okay? So what we do is we put small aliquots of our material. So you might see the little vial that I've got there. You'd fill that with liquid. It would typically be in the kind of aliquots that you want to be using for treatment. You'd put it in here. And what you've got inside here is shells. Hopefully you can see those shells have incredibly cold liquid flowing around them. So what that can do is freeze it. So the part of the freeze part of the freeze drying's done. Okay, so you get a frozen mass, and then what you do is slightly increase. So the kind of temperatures we're looking at, it's probably about -50 roughly. We bring the temperature up slightly, maybe about 4 or 5 degrees. It's very protein dependent, and apply a vacuum. By applying a vacuum, what we can do is we do something called sublimation. Okay, so what sublimation, if you remember your chemistry classes, is that we take the ice, put it to gas, and then it goes back to ice on the coils at the bottom. Okay, and what you're left with, very uninterestingly, is typically a white solid. Okay, a white lyophilized solid, and that solid can then be probably kept on a shelf for years. I'm not sure years, but certainly a reasonable long amount of time. So then that is your, so those by, you then bring the temperature up to ambient, and you'd have a freeze dried solid that the clinician could resolubilize with water for injection and use. So in terms of, if I just take you back to me, I don't know how much I need to tell you about kind of how I'm teaching. I hope that's kind of been done by people like Brenda yesterday. But in terms of your exposure, there will be things like the basic skills and practicals where you get opportunity to get hands-on experience of things like the fermenters that I've shown you, the homogenizer, the distack centrifuge, the filtration kit, and the chromatography kit as well. So that's an opportunity that you get in terms of, depending on, I think it might typically be in your second year. And there's also opportunities to do projects on the kind of kit that I've shown you in things like the pilot plant weeks, which are part of the syllabuses that people do as well. I don't know, I realize it's been a very much a whistle-stop tour of the facilities, and I'm kind of used to, I don't know how much I'd usually try to make a tiny bit more interaction if I've got people in front of me, but I'm very happy to take any questions that people.
Sorry, Gareth, you've just frozen there for a moment. Oh, that's a shame. We've lost Gareth just as we're coming to questions. I'm really, really sorry. It looks like the Wi-Fi has let us down, but fortunately, at the end of the session, if has anybody got any questions that you want me to put to Gareth, just in case we get the Wi-Fi back? It's always the risk with a live event. So we haven't had any questions come in. I do understand it might be because it's, that was a very, very intense, quick whistle stop of a big facility that does a lot of work. So if there's anything that you want to know about, if you'd like to know more about what we do, our engineering, what I'm going to be doing, Gareth, sorry, you froze there for a moment. Just waiting for Gareth to log back in again. So his audio is just reconnecting. So just wait for you to get.
Okay, can you hear me now? Sorry.
Yeah, absolutely. Just to let you know, Gareth,
Apologies
there are no questions that have come through. I think you've covered everything. But but what I was just saying to the audience is, over the summer, we're going to be doing some sessions with our students. So they'll be talking about the kind of things they do, their perspectives on it. They'll be talking about things like Pilot Plant Week, research projects, and the kind of stuff they do. They'll be talking about iGEM. So there's many more opportunities to talk about what it's like as a student to be using the pilot plant. But I really hope that you've got a feel for the layout of it, what goes on there, and really why it's such an exciting facility. It is unique. No other university in the UK has a pilot plant to be able to test and demonstrate and run this. This is actually a scaled down, fully functional facility. So all the theory, all the process that Gareth took you through, all the facilities, they are a scale version of what you might deal with in an industrial setting, which is why when you're taught biochemical engineering at UCL Biochemical Engineering, you're taught with biochemical engineering processes. That's why it's so exciting. That's why our students and our researchers find it such a valuable resource. Okay, we're coming up to the end of time. I don't think any questions come in. Like I said, if there's anything that occurs to you afterwards, you can get in touch with me. You've got my email address. I'll make this available online as well. But I just want to wrap up by saying a huge thank you to Gareth, because it is Pilot Plant Week at the moment. So it's a big ask for him to come in and do this, but I really, really appreciate it. That was fantastic. Thank you very much.
Okay.
Have a great day, and I just want to wrap up by saying I really hope we get to see you in person in future at UCL Biochemical Engineering in London. Have a great day.
Likewise. See you soon.
Bye-bye.
Bye.